All right, good afternoon, everyone. We have a great audience for today. My name is Pam Coons, and I'm the Chief of GI Medical Oncology, so thank you for coming. And thank you for coming to Grand Rounds. It's really a pleasure to host Dr. Elizabeth Jaffee today. So just by way of background, this is the inaugural lecture for the Susan and Dan Koenigsberg Lecture in honor of Dr. Koenigsberg's wife, Susan, and he is here in the audience today. So we are grateful for the gift that has endowed this lecture. So I'll introduce Dr. Jaffee. So Dr. Jaffee is the Dana and Albert Cubby Professor of Oncology, Deputy Director of the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins, and the co-director of the Skip Virag Center for Pancreas Cancer and Associate Director for the Bloomberg Kimmel Institute for Cancer Immunotherapy. She is really an international leader in the development of immune-based therapies for pancreas and breast cancers. She graduated magna cum laude from Brandeis before getting her medical degree from New York Medical College, and then went to get her medical residency in Pittsburgh at the Presbyterian University Hospital, and then finally made her way to Johns Hopkins, where she has been really for the last 30 plus years. And really since her arrival at Hopkins, she has become a really renowned international recognized researcher and in pancreas immunology, and really as close as I know to a GI oncology rock star. So it's such a gift to have her here today. We look forward to hearing about the cancer vaccine era has arrived. So Dr. Jaffee, thank you so much.
I hope I could live up to that introduction. I'm also honored to be the inaugural speaker for this very important lectureship, so thank you very much for this honor. So you heard I'm going to talk about cancer vaccines. I've been doing this for quite a while. These are my disclosures, quite a few, happy to discuss them at the end of the talk. So since this is Grand Rounds, I'm told I have to give you learning objectives. So the first is to understand the current biological challenges in developing vaccine-based immunotherapy for the treatment of cancer. The second is understanding the current developments in vaccine-based immunotherapy testing in patients with cancer. And also we want to understand the current status of vaccines being developed to intercept early cancer development. And this is an area that I'm very excited about. Now, I also like to start out with a little bit of history, love history, particularly cancer vaccines. And so I thought I'd tell you a little bit about what we know and when this all started. So I think we all think about cancer vaccines as starting with William Coley, who was a surgeon in New York in the late 1800s. But actually, the first person to identify that the immune system actually can see cancer was Rudolf Virchow, a German pathologist who observed this probably in the early 1800s. And then we got William Coley, who, I don't know if you all know about him, but he was really remarkable too. These are all amazing physician scientists, by the way. This is what research was in those days. And without these observations, we wouldn't be here. But William Coley, basically what he did was he was able to show that in patients who had subcutaneous tumors, occasionally they'd get super infected with a bacterium or a virus, and those would spontaneously regress. So this really started the early version of cancer vaccines, and he started mixing tumor cells with BCG or other things to get activation. So really what he realized was, infectious agents were probably an innate immune stimulator or adjuvant. And then there was sort of, people tried to do this, didn't really show much activity in patients by mixing these tumor cells or injecting bacterium into them. And then we get to our modern day, which is like, 1985 or so. And why are we able now to move forward? Well, IL-2 is sequenced. IL-2 is a growth factor for T cells. So this allowed different investigators to isolate tumor-infiltrating lymphocytes and show that they can recognize tumors and even prove by adoptive therapy that you could sometimes get regressions. So T cells seem to be very important. And then around the late 1980s, early 1990s, we now have technologies where you can use genetics or proteomics, a lot of work. This is pre-sequencing, but you're able to identify these tumor-associated antigens and be able to use these in vaccines. So this is where my group started looking at genetically engineering tumor cells to express cytokines, the tumor cell being the delivery system for antigens, but others were taking these tumor-associated antigens and pulsing them onto dendritic cells. And then we also had viral and bacterial vectors that were genetically engineered to deliver these antigens. But as things move forward, they still weren't showing much response in patients. We now have better vaccine platforms, such as mRNA, but I'll be honest with you, I don't think it's necessarily the platform. And in fact, I think we now understand that immune checkpoint agents are really what have led to our ability to really make progress with the immune system in fighting cancer. But importantly, also, they're needed for vaccines. And so it wasn't until 2011, CTLA-4 antibodies were approved, and then 2014, anti-PD-1. But also important is the antigen diversity. So sequencing technologies really only became available after 2000 when the human genome was sequenced. And then finally, we now have whole exome sequencing where we can actually look at a person's individual neoantigens and they could be delivered. But again, as you'll see, I think that what was missing for vaccines in all those years was having immune checkpoints, and we'll talk a lot about that. But so immune checkpoint agents have led to this incredible time, right, in anti-tumor immunity. The problem is only 20% or so of patients have naturally induced T cells by their tumors. And that's a real challenge, right? And this is why vaccines are so critical. Vaccines hopefully will be able to induce a quality T cell that can then be activated by these immune checkpoint inhibitors. So that's where we're going. And I work in one of the cancers, of course, that is known to be highly aggressive and non-immunogenic. And for those of you who don't work in pancreatic cancer, about 20% of these patients are eligible for surgery. But unfortunately, only 40% benefit from the surgery long term, 60% will still recur. So we still have a lot to do. And then there's also 80%, the other 80% that have metastatic disease at the time of diagnosis or locally advanced. That are not curable. Immune checkpoint blockade has not worked. Even doublets have not worked. And unfortunately, when you look at these tumors, you may see T cells, but they're ineffective. And most patients don't have effector T cells. They have Tregs and other kinds of T cells, but not effector T cells. Well, why is pancreatic cancer so aggressive? Well, we are able at this point to understand many of the signals within a pancreatic tumor that it just halts the immune system. These include immunosuppressive molecules that everyone knows about now, but it also includes a lot of different immune suppressor cells. So we know about myeloid-derived suppressor cells, tumor-associated macrophages. They're there from the start, and they definitely correlate with more aggressive disease. Tregs as well, but cancer-associated fibroblasts, which I won't be talking about today, but we've been publishing a fair bit on this, they come in many different flavors. We used to think that these CAFs, these cancer-associated fibroblasts, were innate. You know, they didn't really do much. They didn't signal. They were just physical barriers, but they're not. And in fact, if you look at this is a human tumor, human pancreatic tumor. Most of the tumor cells make up about 20 to 40%, whereas the rest of it is the stroma. And the stroma actually houses the inflammation. There's inflammation there. It's just inhibiting T cell activation. And so CAFs now come in multiple different flavors, and they all signal. We're learning that they signal to monocytes, they signal to tumors, they signal to T cells. So they're a very important part of why we have immune suppression. Okay, so we've been testing for quite a while now the concept that we want to reprogram these immune cold tumors, but it will take two steps. The first is to induce a good T cell. It has to be a good quality T cell that can get into the tumor and function in the tumor. So that's the first step. The second step is to optimize T cell function, and that's where the immune checkpoints come in. But what we're finding in pancreatic cancer, which I'm going to talk about a lot, is that the immune checkpoints that inhibit T cell blocking or T cell stop signals, such as PD-1, LAG3, TIM3, are not enough in pancreatic cancer. To make a quality T cell, we also need to hit or activate these agonist signals. And I'm going to talk a bit about 4-1BB. And actually, Li-Ping Chen was part of the original discovery of the 4-1BB pathway, and he's here at Yale. So we've been developing initially these whole tumor cell vaccines. I'm just going to mention them because our initial studies, which are showing progress now, were with these whole tumor cells. And the idea, and this was back in 1989 to 1992, we genetically engineered tumor cells, either autologous or allogeneic, to express GM-CSF, granulocyte macrophage colony stimulating factor. And what we found that this did, and a lot of data was coming from other groups as well, is that GM-CSF recruited dendritic cells, and those to me are really the conductor of the whole immune response, right? These cells know what antigens to take up, process, and present. We know this from all the infectious disease literature, and then they activate both a CD4 and a CD8 T cell. And so this was a collaboration with our colleagues up at MIT, Glenn Dranoff and Richard Mulligan, and at Hopkins was myself, Pai Levitsky, and Drew Pardol. And so we published a lot on this both preclinically and clinically. And my first clinical trial in pancreatic cancer was with 2 allogeneic lines that I developed and genetically engineered to make GM-CSF. And we actually completed a phase 1 study in 2000, we published, 2001. And we were able to show that in the adjuvant setting, we actually had correlation of a very crude evidence of immune response. This was delayed type hypersensitivity reactions to a patient's own tumor cells, pre-treatment and post-treatment, and these are T cells coming into their tumor cells given under the skin. That correlated with disease-free survival, and we have now a group of patients who have disease-free survival greater than 15 years. And when you talk to them, they don't even think they have pancreatic cancer anymore, which is really, that's a miracle to me. So it makes me so happy. We also were able to identify the first tumor antigen, and we did this using serial analysis of gene expression, which came about at that time. And our colleagues at Hopkins, Bert Vogelstein and others, shared their technology with us, and we were able to differentially express genes that were in the tumor cell relative to normal cell. And then we used our T cells, which we were able to isolate from the patients who showed these DTH responses. And with this, we developed an approach to be able to identify tumor antigens. And actually, the first one was mesothelin, which is a tissue-associated antigen, but it actually is a target. And in mesothelioma, it's a particular target that seems to have clinical benefit. Mesothelin as a target alone in pancreatic cancer has so far not shown to be as helpful, although we see T cells against them quite regularly. But we were able to use mesothelin as an example of how giving an allogeneic cell line allowed dendritic cells to take up mesothelin and present it to both CD4 and CD8 T cells. So in 2004, we had the first evidence of in vivo priming in humans, and this was critical. Dendritic cells can process and present on both class 1 and class 2 to activate both types of T cell responses. So moving to now, we've been doing these neoadjuvant and adjuvant window of opportunity platform studies, and the goal here is to identify the best combination to take forward to hopefully eventually FDA approval, because this is a hard disease. So we don't want to be doing big studies. So we decided to do small studies, but do deep dives and try to understand what's going on in the tumor pre-treatment and after treatment. And so these are 20 patients per arm. Our first study we actually reported in 2014, started in 2008. It took a while to get there. Neoadjuvant studies were not as simple in those days, but working with our surgical team, they agreed to wait 2 weeks before patients went to surgery. So we basically treated initially with our just vaccine, we call it GVAX, 2 weeks before patients went to surgery, and then we got their tumor. So we had a biopsy, and then we had their whole tumor. And then they'd go on to have vaccine, adjuvant chemotherapy, and then vaccine until they recur. And so the first arm was completed and reported, and I'll show you results on that. This was vaccine alone. But what we saw was that PD-L1 was going up when we gave this vaccine, and I'll show you some of the immune responses in those tumors. So our second arm was anti-PD-1 plus vaccine. What we found when anti-PD-1 was added to vaccine, now what we started seeing is this signaling pathway CD137 or 4-1BB goes up on the T cells. That was saying to us something like, these T cells are hungry. They're ready to be further activated. And so we came in with our third arm, vaccine plus anti-PD-1 plus anti-CD137. We now have 6 arms, but I have full data, and we've published or about to publish on these 3 arms. So this is our team: surgeons, medical oncologists, computational biologists, really a wonderful team pathologist. So it's a great group, biostatisticians. And so this was the first study, which is vaccine alone. Again, pre-treatment biopsy, vaccine, 1 dose, 2 weeks later, we get their tumor specimens and analyze. And so this was the first observation, which was really surprising to us. And we reported this originally in 2014 when we only had single antibody immunohistochemistry at the time, no multiplexing yet. But we were seeing these lymphoid aggregates coming into the tumor in the vaccinated, but not the unvaccinated. In the unvaccinated, we might see it around the tumor, but not infiltrating. And when you look at anti-PD-L1, we saw anti-PD-L1 going up on the monocytes within these lymphoid aggregates. So when we looked at 39 patients, 33 of the 39 had these lymphoid aggregates, whereas when we looked at unvaccinated, we didn't see any of these that look like sites of activation. We published on this. There's clearly sites of activation. So this was the first description. And actually, if we saw these lymphoid aggregates, it seemed to correlate small studies with better outcomes. So since then, we now have single cell resolution spatial distribution analyses. We have imaging mass cytometry, which allows us to look at up to 40 or 50 proteins at once. And so this is our team. Dimitri is leading this. He's an early career faculty member who's cross-trained in computational biology and genetics. One of my fellows with Alana Fertig, a computational expert. We co-mentored Dimitri. So what he did was he took a look at some of these lymphoid aggregates in our tumor specimens. And what he found was that we have different forms of them, and we've seen this as well in liver cancer. Now both are published. And what we found was that there were those that had necrotic areas. Interestingly, this was only on our triple therapy, which I'll show you in a little while. But many of them had these mature TLSs with true germinal center-like structures, so B cells in the middle, T cells on the edges, and then there are also the ones that are resolving. So when you look at this whole tumor specimen, you see all of these happening all at once, 2 weeks after treatment. So using unsupervised machine learning, he identified a particular signature that he found in these germinal centers of the tertiary lymphoid structures in the tumor. And so what you're looking at here, this is the important part, is that you're looking at just acinar cells, which is in the pancreas. You're looking at germinal center of a regional lymph node to the tumor. Then this is a lymph node that's not regional to the tumor. This is normal pancreas. This is a precancer pancreas, pancreatic cancer, metastasis, stroma. And here is the TLS. And I think it's pretty obvious that pattern 9 only shows up in the germinal center in the lymph node and in this germinal center in the pancreatic cancer tertiary lymphoid structure. And when he looked at TCGA data, looking at just pan-cancers and survival, this is on a 15-year survival curve. What he found was that if, in fact, these patients expressed this particular pattern, they had a longer disease-free survival. When you looked at pancreatic cancer in the same data set, and these are usually primary tumors in TCGA data set, again, 5-year survival seemed to be improved, although let's face it, this is only 8 patients, so it's a very small group of patients. But pattern 9 seems to include 304 genes, and they're all immune-related and inflammatory, so something we're following up on. So to summarize this first part, a lot of this is published. There's much more data out there now. Vaccines clearly can turn even a cold tumor into one that appears to be immune cell rich, and it looks like an immunogenic tumor that now may be susceptible to immune modulating agents. These TLS formation alone, though, does not necessarily correlate with improved clinical outcomes, although sometimes they do, but usually not. This pattern 9, transcriptional pattern 9, may be something that we should follow up and look as we give more and more of this immunotherapy. Are these patients developing this pattern, and do they have improved progression-free survival? And of course, pattern 9 may also correlate with evidence of T cell activation. So the next question was, how are these TLSs shaping T cell responses in pancreatic cancers? So again, going back to our first arm of the neoadjuvant study, this is arm A, vaccine only, biopsy, vaccine, 2 weeks later, we have the tumor, and now we take a look at the intratumoral immune cell infiltrates. And so what we're seeing here are all the different subpopulations of immune cells, from T cells and NK cells B cells and dendritic myeloid-derived suppressor cells. And so what you're seeing here, and both of these are showing that the CD4 T cells and CD8 T cells go up post-treatment. That's red. Post-treatment, pre-treatment is blue. However, when you look at activation, CD8 granzyme B T cells, they're not going up. So for the most part, we're not getting very good activation of these cells. We're just getting proliferation. Something's happening. They are antigen experienced, many of them, but they're not fully activated. When we start to look at comparing anti-PD-1, the second arm, we add anti-PD-1 to the vaccine versus vaccine alone. And remember, PD-L1 went up on these TLSs. That's why we added anti-PD-1. So now what we're seeing is something different. So if we look at the tertiary lymphoid areas and we look at the number of cells in each area, regions of interest, this is increased anti-CD4 T cells in the red bars. We're looking at overall survival for vaccine alone, less than 2 years, greater than 2 years. So we do get a few people that do respond, but it's clearly quite different when you look at the combination treatment, overall survival, 2 years or greater. And we're looking at the T helper 1 to T helper 2 ratio in this case. And those T helper 1s are the ones that are going to activate a good cytotoxic T cell. We also see that there's reduced exhaustion, both CD4 and CD8 T cells. So again, only in the combination therapy, these are CD4 T cells, less exhausted T cells. Same thing for the CD8, PD-1 positive T cells. So that was good news. And then the other thing that we don't spend a lot of time on yet because we don't fully understand, but we see when patients do well and have long-term survival, what we see at 2 weeks post-treatment are these CD17 T cells. And those of you who understand immunology very well know that they're often responsible for autoimmunity. We don't see autoimmunity anymore more commonly in pancreatic cancer with immune checkpoints than we do with any other cancer. We do see it, but not any more so. But these CD17 cells seem to have some role we don't fully understand yet. We're trying to better understand them. But here's the key. So CD137 goes up on these CD8 T cells, and that was critical. So again, looking at these lymphoid aggregates, overall survival less than 2 years, greater than 2 years for vaccine alone. So few patients do have it going up. And then the combination, certainly there's a difference between less than 2 years and greater than 2 years. When you look at CD8 T cells with granzyme B, so a molecule that suggests they're killing the tumor, there is a difference between the CD8 low and CD8 CD137 low and CD8 137 high. So it seems that many of the cells that have that molecule granzyme B are the ones that are CD137 high. And one of my colleagues, Lei Zhang, he did an animal study looking at vaccine alone, vaccine plus anti-PD-1, and vaccine plus anti-PD-1 and CD137, or vaccine plus CD137, and found that it's the triple combination that prevents these tumor-bearing mice from progressing with these KRAS-expressing tumors in the KRAS mice, which is a pretty immunosuppressive mouse model. So it appears CD137 is a CD8 T cell activation marker, and it's an agonist pathway. So it really needs to be engaged to get fully activated T cells. So what else do we know about 4-1BB or CD137? Well, we did a study and reported in 2014 in the HER2/neu transgenic mouse model where we developed TCR transgenic T cells specific for the HER2/neu CD8 epitope. And we had high avidity and low avidity T cells. And what we found was that we could turn these low avidity T cells into functionally high avidity T cells by giving not only our vaccine and anti-PD-1, but hitting it also with 4-1BB agonist or OX40 agonist. And it reduced apoptosis in these T cells, so they stayed around longer. It increased activation of the CD4 T cells, and it also increased tumor killing by the CD8 T cells. And these are just the pathways you could see where naturally APCs are providing that signal 4-1BB ligand to 4-1BB on the T cells. However, you can bypass the APC doing it by giving an agonist antibody, and that's what we're trying to do. So arm C, so this is the arm that's really given us the most information. And we used urelumab originally, and this is the BMS molecule that ended up being taken off the market. It had activity, but unfortunately, it also had hepatic toxicity. They were willing to give it to us, but we had to use a pretty low dose in our neoadjuvant study, and they only allowed us to do 10 patients. We didn't get the full 20 patients. But again, biopsy, pretreatment biopsy, then we get vaccine plus nivolumab, anti-PD-1 plus urelumab, which is the anti-CD137. Then 2 weeks later, we get the tumor mass, and then we go on to package the vaccine plus adjuvant chemotherapy. And for the first time, just 2 weeks later, 1 dose of the triple combination, we start to see pathologic responses, complete pathologic responses in 3 of 10 patients, and additional partial pathologic responses in the others. And this seems to be correlating with disease-free survival. This was the cutoff that we made at this point in analyzing the data. We haven't done a subsequent analysis yet, but so far we seem to see a correlation with the triple combination and patients having longer disease-free survival. And so what are we seeing in these patients? Well, what we're seeing is that when you take a look at the CD8, CD137 positive T cells, it's arm 3 that has the most of these, so the triple combination. When you look at CD8, just granzyme B, so not CD137 positive, just granzyme B CD8, all 3 vaccine combinations have them. But if you look at CD8, CD137 granzyme B T cells, only the triple combination shows it. So it seems like the CD137 T cells are quite critical. So to summarize this part of the talk, so far what we know, if you take a look at this is our lymphoid aggregate in the tumor microenvironment. You give vaccine, and what happens is we get T cell activation. We're seeing that, but it's weak. It's not doing a lot for the patient. We add anti-PD-1, and we get enhanced T cell activation. We get CD4 chemokines and cytokines being upregulated, and we get upregulation of CD137, particularly on the CD8 T cells. And then we give the CD137 agonist, and now we're seeing increased T cell expansion, increased T cell motility, increased T cell avidity, and reduced exhaustion. I'm not going to show you all that data. These are not antigen-specific T cells. These are just the T cells we're finding in the tumor. We just published on that as well, because I want to show you more data on what we're seeing now with our KRAS vaccine. But I can tell you that we're now enrolling. We've enrolled our 4th patient onto a new cohort, and this is a collaboration with Agenus that has a 2nd generation anti-CD137, and this is what our study looks like. We're comparing our vaccine GVAX, the whole tumor cell, with Agenus and anti-PD-1 versus our KRAS vaccine, which I'm going to talk about now, with the same combination in the neoadjuvant setting. Eric Christensen, a young faculty member, is leading the study with us, and this is ongoing. We had to do a dose escalation with the Agenus 2373. We've seen no increased toxicity. All of the patients made it to surgery, and so we're now enrolling on to the therapeutic dose of this agent. So emerging role for neoantigen cancer vaccines in immune-resistant GI cancers. This is really an exciting time. I'm very excited about it. I know talking to many of the people last night for participating in these studies, they're also excited. So why now? Why is now the time? Well, I think having these next generation technologies where you get a biopsy and within a week, you know what the neoantigens are, makes it much easier to begin to tailor these vaccines to patient-specific neoantigens. Also, we can test them in early stages of disease and have a readout, right? So ctDNA now allows us to monitor if patients are responding, and you can monitor patients in pancreatic cancer once they've had the cancer. Often we can detect when it's coming back. Unfortunately, you know, by the time you detect it, we know it's coming back pretty quickly. But still, we have a mechanism of looking at that, which we didn't have before. And we could also move to the neoadjuvant setting and do these multi-omic type analyses, which give us a lot of biology and helps us make our treatments better. And then I think we can do these combination therapies, and we have new vaccine platforms as well, the mRNA delivery systems. Although I'm going to be honest, I don't think we've made a head-to-head comparison of all of these delivery systems. So I'm not sure it's all about the delivery systems as much as what are we giving with the delivery systems and what are the antigens we're putting into these delivery systems. So what are the key components to successful non-virally driven cancer vaccines? Well, the first is really selecting the antigen. It sounds simple, but it's really not. I think anyone who's done this, we know that the current platforms available for picking neoantigens, even when you get a list of 10 or 20 that the patient's tumor expresses, That doesn't mean the immune system sees them all or is able to induce a quality T cell. We don't have a good way of looking at that yet, because really the best way to look at it is to know when the TCR binds to the MHC epitope, because it's a conformational change, not the avidity of the MHC epitope or the overall avidity of the T cell itself, but the interaction. And that's where we don't have data sets yet. We need those data sets. We also need to optimize adjuvants, and I think some of these new platforms may be the way, but we do have better adjuvants today than we had 15 or 20 years ago. They target innate signals like TLRs, so that's important too, and then obviously the platforms. But most importantly, we need to optimize T cell quality, and how is that going to happen? Well, that's only going to happen if we are able to get the right immune modulators. And then we also need to target the right timing, the right patient populations. Metastatic disease, we are seeing some responses, but clearly earlier stage disease is critical. But I do believe vaccines are important because what do they do most of all? They induce memory. So even if you combine them with other agents, it's the vaccine that's going to provide the memory responses. And that comes in both CD4 and CD8 T cells. So CD4s are very important. Okay, so neoantigen targeted vaccines. So they come in different flavors too. So we have the shared neoantigens. Mutated KRAS and pancreatic cancer is one that we're spending a lot of time on lately. I am, others are as well, and there's a lot of exciting clinical studies out there on this. And then there's the personalized neoantigen vaccines. Well, the personalized vaccine pipeline, think about it. So you need a biopsy. You also need the PBMCs to be able to figure out the neoantigens. Then if you're lucky with whole exome sequencing and RNA-seq, 1 to 2 weeks, maybe 3 weeks, depending on who's doing it and how fast they can do it. And then you have to predict, and that's where the problem comes in. I mentioned we don't have the best predictors of what neoantigens are going to be the best immunogens. And it's not about binding. Some of the least binders, the lowest binding neoantigens are the best antigens. In fact, KRAS doesn't even come up on the list very well. So it's really an area that if any of you are working on this, please, it's going to be very helpful to us. And then you need to generate your vaccine, and depending on the vaccine depends on how long it takes. So what are the key considerations? Time for vaccine generation. Pancreatic cancer, you want to have a couple of different types of vaccines because a neoantigen vaccine may be hard to do on every patient. Also, pancreatic cancer patients don't have a lot of neoantigens. So some will, and those are the ones you want to try this on. But again, we need to think about this more broadly so we could really help most patients with pancreatic cancer. The cost may limit broad-scale production. I mentioned the predictive algorithms, small subsets of predicted neoantigens. And again, I would tell you that the majority of our patients have low mutation burdens. So with that said, though, we have some really great studies out there that have shown evidence of activity both in pancreatic cancer and in melanoma and hepatocellular carcinoma. So personalized RNA vaccine in melanoma, I'm very excited to see what the phase 3 will show. These are neoantigens given with an anti-PD-1, Pembro. And so the phase 2, randomized phase 2 looked very promising. Personalized RNA vaccine for pancreatic cancer, I think there are people here who are participating in the phase 2. The phase 1 was a small study, but it looked very promising as well with anti-PD-L1. And then I was part of a hepatocellular carcinoma vaccine, which was a DNA vaccine, Genios, run by Mark Yartrin at our institution. He was the national PI as well. I was not hopeful for a DNA vaccine, except for the fact that it also gave IL-12, which to me is one of the best adjuvants that's out there. So the DNA vaccine gave IL-12 in a DNA construct. So IL-12 systemically is quite toxic. And then there's the Amplify, which is the Alisio Therapeutics vaccine. And that's a peptide vaccine targeting KRAS, just 2 different mutations in KRAS. And their mechanism is it goes directly to the lymph nodes. So maybe that's a good way of getting the best T cell response. So, but why KRAS? So we've been very excited about KRAS. Well, KRAS is probably the most important mutation in the majority of pancreatic cancer patients, and it happens early. It's the earliest known mutation in pancreatic cancer, and that's why we're very interested in it, because not only may it be good for therapeutics, because if you get rid of KRAS, the tumor can't survive. The tumor depends on KRAS, but also can we eventually go to high-risk patients? And I'm going to tell you the answer is I think so. But anyway, so KRAS, the other nice thing about KRAS is there's only about 6 or 7 mutations that cover all of the mutations of pancreatic cancer, easy to make a vaccine. Also, I don't know if you're aware of this, but my colleague, Laura Wood, has published that anyone over 50 has probably 100 precancers called pannins. These are in healthy individuals. And interestingly, they don't express one mutation. They'll express multiple KRAS mutations. Now, by the time they become a cancer, in the few patients who go on to get cancer who have these pannins, one mutation will be expressed. But if you're thinking about prevention, you may want to be targeting multiple mutations. So KRAS causes chaos in pancreatic cancer, if those of you who haven't looked at it. So it actually is responsible for a lot of the immune suppression. It causes PD-L1 going up, which prevents T cell activation. It causes TGF-beta to be expressed, which turns CD4 T cells into Tregs. It attracts and co-opts myeloid cells to become MDSCs and tumor-associated macrophages. And it also interacts with CAPS, cancer-associated fibroblasts, to prevent CD8 and CD4 T cells from trafficking. So it's a problem. We also know that KRAS can be a target because one group, Steve Rosenberg's group, had a patient with lung metastasis. They were able to isolate TIL, sequence the TCRs, put the TCR of interest, HLA-AC 0802, that saw KRAS into allogeneic lymphocytes and give it back. And one patient had regressions of their tumor, and they expressed, obviously, HLA 0802 KRAS. So it is a target. It can be used for lysis. So we have a big team. Our team has surgeons, medical oncologists, immunologists, pathologists, PhDs, gastroenterologists, et cetera, and a lot of trainees, computational biologists. And we've been working on developing our KRAS program. So the first I'm going to tell you about is our vaccine for the treatment of adjuvant pancreatic cancer. And so we developed 6 long peptides, 21 amino acids, with each of the 6 mutations that are the most common. Every patient gets all 6 peptides mixed with poly-ICLC, which is a TLR3 targeted innate stimulator. And we've done it now in resected PDAC, metastatic colorectal cancer. And I'm going to show you also our interception study, which we completed. So this peptide we gave in the adjuvant setting. And again, even in the adjuvant setting, vaccine alone is not enough in pancreatic cancer. We gave it with anti-PD-1 and anti-CTLA-4. This is the BMS, nivolumab and ipilimumab. And so we gave priming. Patients underwent surgery, had their chemotherapy, and then got enrolled. So this is true adjuvant. And these patients got priming doses and then boosting until recurrence. And so we monitor right out of the patient overnight stimulation with their peptides. I want to see as close to the patient as possible, not a long in vitro stimulation, an interferon gamma ELISpot done in our clinical immunotherapy lab. And we quantitate just the number of NKRAS specific T cells pre-treatment and post-treatment. We do it over time. So we have a time course of how they develop their KRAS specific T cells. We find that they peak at 17 weeks and then they kind of level off. Then we do flow cytometry or mass cytometry to look at all of the T cell populations and to profile their activation and memory status. And we also do single cell RNA and TCR-seq to isolate these TCRs and look at what they look like. And so this is under secondary review. Amanda Huff is a PhD postdoc who's now on faculty, who's worked with me on this and developed how we do this. And so what you're looking at here is the maximal relative fold change from baseline to 17 weeks. Each line is a different patient. We did 12 patients because we wanted to get to the high-risk population. So this was more of a safety and immune induction study. And the blue are the patients who had an increase in KRAS-specific T cell responses, best response. But then we looked against each of the individual mutations. Each line is a patient, pre-treatment, and week 17. And I think it's pretty obvious that it didn't matter what their mutation was in their tumor. G12V responses and G12R and G12C are the best responses. G12A, okay, G12D, not so great, and G13D, not so great. And so we started to see a pattern here where G12D and G1213 D are really the problems in this, at least in this small study. So far, we're seeing some decent disease-free survival based on maximal fold change, the average KRAS response to any epitope, the best response. And if you're at the 25th percentile, high 25th percentile, this is maximum fold increase, just all comers on the study. And then this is looking at the T cell responses to their specific mutation. So if you had a mutation in G12R and you responded, you were great. It didn't matter that your tumor expressed it. I'll be honest with you, G12D, not a good thing. So those are the patients that seem to recur. And this is a different way of looking at it. So what we're looking at here is patient ID, each individual patient on the y-axis. This is the number of positive mutation responses. The majority had it to all of them, but there are a few patients who did not. And then if you look at mutation-specific response, so these are the 4 G12D patients, you could see maximum response to G12D peptide, not so great, although they responded to the other peptides. G12R patients did very well, and G12V patients did very well. And then this is just looking, we do it over time. This is just to 17 weeks. You could see it's going up. And again, G12V being the best, G12R being second best, just overall responses in patients. I'm not going to go through all this data just to tell you that with CyTOF profiling the types of T cells and their activation and memory status, really what we're seeing by 17 weeks is that we can see a really nice CD4 effector and central memory response and CD8 effector memory response. And again, this is work of Amanda's along with Wanjin Ho, who's an associate professor at this point at Hopkins. So now Amanda wanted to do TCR-seq. So the way she did this was this did require a 7-day stimulation with each individual peptide, and then we do the TCR beta sequencing, and then we map them back to the TCR alpha chain. And so what you're looking at here are the 4 patients we did complete TCR sequencing. This is the fold change relative to control, and just looking at the number of TCRs against each of the individual peptides. So this patient did very well, this patient did very well, this patient, not as many TCRs expanded, this in between. When we look here at the patient ID, the 4 different patients on the y-axis, when we look at the TCR expansion, these are the individual TCRs now, you can see the different colors are the different epitopes. So of course, G13D and G12D are the least expanded even in these 4 patients. What we found was that there were also some cross-reactive. In fact, up to 25% of the TCRs saw more than one epitope, and these were CD4 TCRs, so we're not that surprised, but it's still very interesting when you think about just maybe there's some evidence of cross-reactivity, epitope spreading, those sorts of questions. When we just map the KRAS TCRs onto the data of the expansion and the activation status, what we found was they mapped very well to CD4 T central memory responses and CD8 effector memory responses. So again, correlating with what we saw in just the overnight stimulation and then bulk analysis by CyTOF. When we validated, we do see T cell recognition of the antigen and killing. And this is just an example where we have a Jerkit's GFP reporter system, and then we transduce into these TCRs, individual TCRs, and then look at killing of both the epitope pulsed onto tumor cells and the tumor cells expressing the right MHC. And what we find is that most of them do kill and they're single, but this is an example of a G12V, A, C, and G13D recognition, and there's responses to all of them. And then interesting, the one other observation was a public cross-reactive TCR that we found, much less common, but we're seeing these are 3 different patients who had the same TCR seeing the G13D epitope. So that's also something that we were not expecting. So there are some public, again, CD4 T cells. Just to show you this approach, same vaccine with Ipi-Nivo, we can see responses in metastatic colorectal cancer. So these are patients that we treated, and we have 3 that had a 30% decrease or greater partial response. But what's important here is that I don't know if you follow any of this data if you're not using these agents, but often they work well in lung metastasis, but not in liver. We're seeing liver metastasis with responses too, and this is one example of response in liver. So again, we have to figure out what is different about the liver compared to the lung. There's some data that's providing us some evidence that's been published in preclinical models, but we need more human data on this. So finally, KRAS inhibitors, game changers, those of you using them, also hard to get for our patients. We spend a lot of time, and I'm sure you guys do who are giving them, finding open trials. But KRAS inhibitors are game changers for many reasons, one of which they alter the immune environment. So we're seeing both currently in animal models, mostly, an increase in MHC class 1 and 2 in pancreatic cancers, reprogramming of these myeloid-derived suppressor cells to become T-cell functioning or T-cell activating agents, an increase in T-cell infiltration. And so far, what's been reported is about a 30% response rate in second-line treatment of metastatic pancreatic cancer. I don't think any of us have seen that before. And also, we see a 70% disease control rate. The problem with these is that resistance occurs in less than 12 months. So we've made some great progress, but now we need to get to that second level. And there's certainly a lot of work being done on the resistance mechanisms. But my feeling is, why not integrate these inhibitors with the KRAS vaccine immunotherapy? Because KRAS inhibitors also make the tumor microenvironment more immune welcoming, I guess is the best way of saying it. But anyway, so we have two studies. We have two new cohorts now, and these are in first-line chemotherapy, best response before patients go on to progress. So it's basically maintenance. We give them after they get to that point, because for those of you who give this chemotherapy, it's not easy on these patients. And so then we give vaccine, and we're now working with Agenus with their anti-PD-1 and anti-CTLA-4. So, and this is ongoing, and we get a baseline biopsy after chemo. We obviously have it before chemo, and then we get on-treatment biopsy. And this is run by my colleague, Neil Azad, who's a KRAS inhibitor expert, and one of our fellows, Kai Leong. And the second one is actually KRAS inhibitors. We can't get it to look at concurrently yet, but our preclinical data suggests that giving it after RAS inhibitor therapy may also be of benefit. And so these patients get a pretreatment biopsy, a biopsy once the RAS inhibitors are no longer working or they're stabilized and they come off therapy, and then we add the vaccine plus the immunotherapy agents. And so these two studies are enrolling and ongoing. And finally, we have moved into the high-risk population. So the big question is, can we intercept? And when do we want to intercept? Well, we want to intercept early at the first mutation. Oops, sorry. And what's important is that from the time of the first transformation to aggressive pancreatic cancer, it's about 10 years. So we have some time. But we really want to get these low-grade pannins. These are the precancers, or these these high-grade cysts before they progress on to pancreatic cancer. So that's what we're trying to do. We're trying to get it early before other genetic alterations occur. And so up to 90% of these precancers will harbor KRAS mutations. And as I mentioned, a single pannin can harbor multiple KRAS mutations. So we want to come in with the 6 different peptides. We've taken a look at these pannins. And so you can't see these pannins typically on radiographically. So basically, the only time we really get to see many of these is within the pancreatic tumor that's resected. And so that's what we did here. We did a study that recently got published where we looked at the tumor, we looked at pannins that were not near the tumor adjacent, normal duct, and chronic pancreatitis, which is often there too. And we did imaging mass cytometry. And basically what we saw was that if you take a look at normal pannin, pancreatic cancer, and chronic pancreatitis, if you just look at CD4 T cells, they're much higher in the chronic pancreatitis and the pancreatic cancer. Even the CD4 to CD8 ratio is still higher there. However, if you take a look at what are those CD4s, you see that most of the CD4s in pancreatic cancer and chronic pancreatitis are FOXP3 positive T regulatory cells. If you now look at the CD8 to CD4 ratio, what you see is that these pannins have more CD8s to CD4 ratios, very few of the Tregs. And it seems like these earlier pannins, these are early stage pannins, do not have mechanisms of suppression. By the way, they already are engaging CAFs around them, that's all in our publication. I don't have time to talk about that. So they are starting to form evidence of, I'm getting ready to become more aggressive. Also important, they have few exhausted T cells. You could see here, very few exhausted T cells, whereas pancreatic cancers and chronic pancreatitis, those T cells are exhausted. So we tested in 20 patients who are at risk, actually 20 healthy individuals who are at risk of developing pancreatic cancer. They could have a genetic reason, they could have family history, they could have a radiographic, but most of them just had genetic or family history. And so we just gave our vaccine with the adjuvant, no immune checkpoints, they're too toxic in their current form. And we did prime doses and then boosting. And we've done all the analysis I showed you on the adjuvant study. And what's interesting is the majority of our patients, and we did 20, you can see here, completed the study. The majority of our subjects on the study, number of positive mutation responses, most of them did respond to all of the mutations. And you could see, again, by 17 weeks, we're seeing them reach their peak when they do reach a peak. Of course, some patients didn't necessarily maintain that peak. And interestingly, the differential immunogenicity of the different mutations isn't as great when you test it in people who are low risk, or who are at risk but have no cancer. So this is, again, each individual person on the study, baseline 17 weeks, G12R, G12B, G12A, G12C, G13D, and G12D. Better responses overall. And then we do see in vitro expansion with ELISpot. And we're able to show that we're able to get number of T cells expanding, and also we see them out for quite a while, so they can sometimes expand 3 different patients. This is at 1 year. This patient didn't do very well. This is a G12R, but this is a G12D patient, and we're seeing great responses at 1 year. And this is a G12V, another one with great responses at 1 year. And at 2 years, we have 1 patient out, and this is just looking at polyfunctional T cell responses. And again, we're seeing at 1 year and 2 years some polyfunctional responses. We did stop the vaccine at 1 year. I don't know if that's the best thing to do or not. We have to learn from this study. So the final study ongoing that's now enrolling is one with these IPMNs. These are cysts that we could see radiographically. We could tell if they're starting to transform. They still have 1 or 2 months. So this is a window of opportunity where they get screened, we get the biopsy, we treat them for 4 weeks with our vaccine, and then we get their surgical specimen. So this will allow us to look at the TCRs that are in the periphery and whether they're trafficking into the tumor. So I'll end there, and I'll just say that immune checkpoint agents have hit the low-hanging fruit. They're exciting, satisfaction that it can be cancer, but not pancreatic cancer, unfortunately, but there's hope for a cancer-free future if we know how to combine them with the right agents, whether they're targeted agents, whether they're vaccines. But I think we need a lot more work to really move immunotherapy to the next level. Cancer vaccines can induce T cells in patients without pre-existing quality T cells. I think our advances in technologies that uncover neoantigens are really making this something that's possible to go patient-specific, hopefully. And we do still need immune checkpoints or immune modulators to help make these T cells work in patients with cancer. I think it's important that we move to high risk or to people at risk. The global cancer vaccine drug pipeline market is great. I think cancer rates are increasing globally. We need to really be thinking about that. The market for therapeutic and prevention vaccines is going to continue to grow. I know it's not going to make as much money, but it's going to be critical if we really want to stop cancer at its earliest stages. And also, I don't know about all of you, but I'm frightened. Early onset cancers are a real thing, and even in 20 to 39-year-olds. So we really need to be thinking about trying to intercept early. I'll stop there, acknowledge all of my partners and grant funding, and this is our team. Thank you.
That was fantastic. Thank you so much. We have time for a few questions. Sure, go ahead.
Yeah.
Yes.
Repeat the
I
question.
was wondering about the site of the inoculation of the vaccine. It seems that you are inoculating the vaccine in the arm of these patients, and I was drawing a parallel with the immunogenic cell death that is observed locally with some ICD inducers. And I was wondering, particularly in the neoadjuvant setting, whether your method would be much more efficient if you would inject, let's say, by endoscopic ultrasound, if you vaccinate the patient by intratumoral or by injection in a draining lymph node, a peripancreatic lymph node.
Yeah, so the question is site of vaccination, and are we really doing the right site? And I think that's a really important question. I think that, doing intratumoral does have some activity. We've seen that with adjuvants. We've seen it with some vaccines. The problem is getting the immune response to really effectively get to where it needs to get. You're just getting into one or two sites. It's not very easy to do. And so the preclinical modeling have shown that the best responses actually occur if you do intratumoral and systemic, or, intradermal or intramuscular. But I think it's an important point. It gets to my point about the method of immunization as well as, the route. I think we don't really have enough information. And the problem is, it's so hard to do this in patients, right? How many patients do you need to do this? It's just really hard, and to make these comparisons and the cost of it. So that's the problem I find. And obviously animal models are not going to tell us those answers well, but it's a really important question. Thank you for asking it.
Jeremy. Hello.
Dr. Jaffee, wonderful, wonderful tour de force talk. Thank you so much. You presented the data showing that the different mutations in KRAS have different immunogenicity, right? And of course, we know that the G12D is one of the worst in prognosis to begin with. Could you comment on why you think there's a difference in the immunogenicity in your vaccine, and whether or not it's intrinsic to the mutation or to the host response?
That's a great question. And I'll be honest, I don't have the answer. What I do have is just little pieces of data that tell me the tumor microenvironment is probably the answer. And what we're trying to understand is if the different mutations alter the tumor microenvironment in different ways. And interestingly, when I showed you the colorectal cancer, a G12D patient did respond. But the colorectal tumor microenvironment is probably very different than the pancreatic cancer tumor microenvironment. And even the primary and the metastases are going to be different, right? We've done studies where in the mouse, where the same tumor growing in the liver versus in the lung respond differently to immunotherapy, pancreatic cancer or colon cancer. And the only differences we're seeing at these sites are the CAPs, and then also the dendritic cells. So the lung, and I think this is why lung tumors respond better. Dendritic cells are there ready to prime because the lung is constantly being bombarded with infection, right? Whereas the liver's protecting, a vital organ. You can't live without the liver. It detoxes. It does so many different things, right? So I think that we really need to understand, even among these different mutations within the different sites, within the different cancers, what are they doing to the tumor microenvironment? And it may be a timing too, depending on what time in the cancer's cycle. Great question. I wish I had the answers. I just have those observations right now.
Maybe one more question. Go ahead.
Thank you. For the first part of your the neoantigen specific vaccine for neoadjuvant setting pancreatic cancer, the anti-PD-L1, anti-PD-1 antibody was used, but for the KRAS specific antibody vaccination study, the anti-CTLA4 plus anti-PD-L1 is used. So what do you think the rationale to use anti-CTLA4, the antibody for the combination of vaccine therapy?
Right. So yeah, we've published a lot on anti-CTLA4. Great question. So what's the rationale for anti-CTLA4? So our original data for that is based on two metastatic studies, one of which was our vaccine plus anti-CTLA4, and the other was vaccine plus anti-PD-1. And we did TCR sequencing. And this was, granted, this was early on, so this was published probably 2020, so it was earlier technologies, but we were able to get really good TCR sequencing. And we had patients who had clinical responses in both studies. So we correlated it with the clinical outcomes, tumor responding, so that kind of clinical outcome. So we thought that that was an important correlate. And so what we found was that the TCRs that were working in anti-CTLA-4 plus vaccine were TCRs that were from de novo T cells. Okay, so CTLA-4 was increasing the number of de novo T cells that were unprimed and getting them to expand. Anti-PD-L1, the patients who did well were ones where they already had good T cells, and the PD-L1 expanded those T cells that were already activated. So we find that the two together are very important. We usually prime with CTLA-4 twice, and then we give anti-PD-1 because obviously the two together is more toxic than individual ones. More recently, we've come up with the CD137. Now we've eliminated, I'm not sure if you were asking that or not, but we did eliminate that in the neoadjuvant study. We just didn't want to complicate things. We want to see how things go. And we think that the CD137 has been quite helpful. And it's earlier stage disease, so maybe the vaccine itself is doing a better job of priming in the earlier, but it's still, these are all open questions, but that's what we know. And then, certainly Jim Allison has shown that CTLA-4 blocking will reduce Tregs in mouse models. And we've seen that in humans as well. Again, published it. And, in pancreatic cancer, Tregs are pretty important. They come in pretty early. So I think that's another way in which CTLA-4 signaling can affect these T cell responses. But great question. Thank you.
Great. Thank you so much for joining us today. A huge thank you to Dr. Jaffee for spending the day with us. I'd like to, a couple other just brief things. I'd like to say another big thank you to Dr. Koenigsberg for sponsoring this lecture. We will have this annually, so we thank him for that gift. And then for trainees who are around, Dr. Jaffee will be spending the next about an hour just in this room behind us. All trainees are welcome. Give us maybe 3 to 5 minutes to just regroup. But thank you all for attending today.